Hello everyone, this is the third lecture of Module 3. We will learn about phytohormone. It's one of the most important topics in plant physiology. Okay, let's start the lecture. Phytohormones are small signaling molecules. Four groups of plant hormones are recognized. For example, auxin, cytokinin, ethylene, and gibberellin. These small signaling molecules regulate plant growth, development, and the responses to the environment. Usually, plants produce very low concentrations of hormones in the micromolar range for auxin and ethylene, and in the nanomolar range for cytokinin and gibberellin. Although each hormone has unique pathways, most share a common mechanism. Each hormone is recognized by a specific receptor. The receptors are localized in various places of cells. For example, auxin and the gibberellin receptors are found in the cytoplasm, and the cytokinin receptors are observed in the plasma membrane. After signal perception, transduction begins by second messengers. This signal transduction modify gene expression and finally induce the cellular responses. Let's learn more about auxin hormones. There are two types of bud in stem apical bud and lateral bud. The apical bud is active, but lateral buds are dormant, and it's the influence of auxin hormone that maintains this state. The shoot tip contains high levels of auxin, and it suppresses the growth of lateral buds, allowing main stem to grow tall. In other words, high auxin in the shoot tip suppresses the growth of lateral buds. However, after pruning or topping, the lateral buds actively grow and become bushier because low auxin concentration promotes the activation of lateral buds. Pruning is a horticultural technique that is the removal of parts of plant to control plant shape, enhance growth, and increase yield. Pruning reduces auxin concentration and it promotes to grow branches actively. Consequently, the plant becomes bushier. It's an important procedure for cannabinoids and seed production. Cannabis growers often use cloning for propagation. Cuttings obtained from the stem are treated with auxins such as IBA to stimulate new root formation from the cut stem site. The white powder you see in this slide is IBA. Adventitious roots are roots that originate from non-root forming tissues such as stems and leaves. There are other effects of auxin. One is phototropism. It's a physiological phenomenon that auxin moves to shade side and stem bends toward light. The other effect is gravitropism, and it's a plant's growth response to gravity. Auxin helps roots grow downward and shoots upward in response to gravity. In addition, Auxin promote fruit growth after fertilization. There are natural auxins and synthetic auxins. IAA is a natural auxin and it stimulates root growth in plants. IBA and NAA are synthetic auxin. They promote rooting in cuttings. 2,4-D is also a synthetic auxin and it acts a herbicide that kills broadleaf weeds by overloading them with auxin. It overstimulates auxin signaling in dicots. However, grasses are more tolerant. Cytokinin is a phytohormone to promote cell division in meristems. The word cytokinin comes from its role in cytokinesis, which is the division of the cytoplasm during cell division. Cytokinin works together with auxin to promote cell division in meristems. Cytokinin stimulates the development of shoot, branching, and leaf formation. As you can see here, high cytokinin of culture medium stimulates shoot and leaf formation from the detached plant tissue, while high concentration of auxin promote root formation. This slide shows the organ formation from the callus according to the difference in the ratio of auxin and cytokinin concentrations. In short, hormone ratio determines organ fate. More auxin means root formation, more cytokinin means shoot formation. Callus is a mass of undifferentiated plant cells that forms at the wound site or in tissue culture. The third plant hormone is ethylene. Ethylene is commonly known as a hormone that regulates fruit ripening, stress response, and aging. However, it has a somewhat different function in cannabis. Let's find out about these in the next slide. Ethylene is the smallest gaseous hormone. Ethylene acts like a female promoting hormone in cannabis plants. So, high ethylene stimulates more stable female flowers, while low ethylene induce male or hermaphrodite flowers. In the previous slide, I mentioned that ethylene maintains female flower formation in cannabis. By inhibition of ethylene production, however, we can produce feminized seeds. When female plants are treated with muscularizing chemicals such as silver nitrate and silver thiosulfate, they inhibit the production of ethylene and that induce formation of male flowers. These male flowers produce pollens, carry X chromosome. Feminized seeds will be created if these pollens are pollinated on female flowers. Feminized seeds have many benefits, so cannabis growers choose feminized seeds than regular seeds. Regular cannabis seeds produce male and female plants. Male plants release pollen that can fertilize females, and fertilized male flowers produce seeds. However, cannabinoid content decreases. Cannabis growers generally aim for unfertilized female flowers to produce high yield of cannabinoids, but even one male plant pollinates the crop, reducing quality and yield. Expected yield may drop by 30 to 50% due to male plants. Lastly, gibberellin, I'd like to mention about that. Gibberellin is a hormone that has many functions, but it has a unique function in cannabis. As you can see in this slide, spraying gibberellin on the leaves induces male flower formation in female plants. This ability can be used to produce feminized seeds in cannabis. Gibberellins are mainly produced in actively growing regions of the plant, such as young leaves, root tips, and shoot apical meristems. Break seed dormancy and promote fruit set and growth without pollination. As we saw in the previous slide, gibberellin can induce male flowers in female plants. This ability can be used to produce feminized seeds. Spraying a 0.01% GH3 solution for 2 to 3 weeks is needed. After appearance of male flower buds, the male flowers produce pollen carries X chromosome. Feminized seed will be created if these pollens are pollinated on stigmas of female flowers. We'll stop here for today. I'll see you next class. Thank you. Bye.